Good day, sir. I am Kathleen Rose F. LAbog from BS Ed. This is my laboratory number 4. My topic is what is mutation and gene regulation. When we say mutation, is an alteration in the nucleotide sequence of the genome of organisms, virus, or extrachromosomal DNA. Viral genomes contain either DNA or RNA. Mutations are changes in the genetic sequence, and they are a main cause of diversity among organisms. These changes occur at many different levels, and they can have widely different consequences. In biological systems that are capable of reproduction, we must first focus on whether they are heritable. Specifically, some mutations affect only the individual that carries them, while others affect all of the carrier organism's offspring and further descendants. The next is the types of mutation. The first is missense mutation. This type of mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene. When we say missense mutation, is a point mutation in which a single nucleotide change results in a codon that codes for a different amino acid. The second is nonsense mutation. Nonsense mutation is the substitution of a single base pair that leads to the appearance of stop codon where previously there was a codon specifying an amino acid. A nonsense mutation is also a change in one DNA base pair. Instead of substituting one amino acid for another, however, the altered DNA sequence prematurely signals the cell to stop building a protein. The next is silent mutation. Some mutations that change DNA bases do not have any effect on the sequence of amino acids in the proteins. A silent mutation is a change in the sequence of nucleotide bases which constitutes DNA, without subsequent change in the amino acid or the function of overall protein. The next is insertion or deletion. Insertions are mutations in which extra base pairs are inserted into a new place in the DNA. Deletions are mutations in which a section of DNA is lost or deleted. An insertion changes the number of DNA bases in a gene by adding a piece of DNA. A deletion removes a piece of DNA. Insertions or deletions may be small, one of a few base pairs within a gene, or large, an entire gene, several genes, or a large section of a chromosome. The next is duplication. A duplication consists of a piece of DNA that is abnormally copied one or more times. Duplication is a type of mutation that involves the production of one or more copies of a gene or region of a chromosome. The next is frame shift mutation. This type of mutation occurs when the addition or loss of DNA bases change a gene's reading frame. A reading frame consists of groups of 3 bases that each code for one amino acid. A frame shift mutation is a genetic mutation caused by indels of a number of nucleotides in DNA sequence that is not divisible by 3. The next is repeat expansion. Nucleotide repeats are short DNA sequences that are repeated a number of times in a row. For example, a trinucleotide repeat is made up of 3 base pair sequences, and a tetranucleotide repeat is made up of 4 base pair sequences. A trinucleotide repeat expansion, also known as triplet repeat expansion, is the DNA mutation responsible for causing any type of disorder categorized as a trinucleotide repeat disorder. And the last one is, what is gene regulation? The regulation of gene expression conserves energy and space. It would require a significant amount of energy for an organism to express every gene at all times. So it is more energy efficient to turn on the genes only when they are required. Regulation of gene expression or gene regulation includes a wide range of mechanisms that are used by cells to increase or decrease the production of a specific gene production. That would be all. Thank you.